Okay, so section 4.4. We're going to be discussing this group of organisms, eukaryotic organisms. They all have a nucleus. They all have internal organelles. These are going to be a group of organisms that just don't fit anywhere else. Turns out we scientists, we biologists, we like to group like with like. We put all the fungi together. We put all the plants together. We put all the animal together. But then there's the leftovers, things that just don't fit. And as long as they are single cell, well, we lump them in with the protozoa. You're going to see with the protozoa, there's no set shapes. There's no set body types or cell types or abilities or environments. They're pretty much everywhere. They're pretty much all different. So the protozoan is just an umbrella term. It's anything that just doesn't fit in the other categories. But it doesn't say we don't want to sit there and categorize the protozoa because we're kind of, retentive. We want to group like with like. So that way when we look at a group, if we know something about one thing in that group, we can kind of understand others in that group. So we have we have criteria. These are the criteria we use to group like with like under the protozoa. When we look at them, we look at the cell itself. We look at how it's formed. Ectoplasm, endoplasm. Not all of them have this ectoplasm versus endoplasm. Ectoplasm, you'll see just below the plasma membrane, this area of, well, pretty much no organelles. And then as you get deeper and deeper into the cell, you start to see it becomes more granular. But you start to see more and more organelles as you get towards the center of the cell. That's the endoplasm. Distinct regions. Do they have it? Yes, no. One of the big criteria we utilize is motility. These are single-cell organisms that have to get from point A to point B. So how do they get from A to B? They're not going to use so much the run and tumble we talked about with bacteria. There's going to be a little bit more going on. Still single-cell organisms, so they can't decide, I want to go at A to B, so here's my direct line. But their methods of movement is quite a bit different. For some of these organisms we're going to talk about that take on an amoeboid shape. It means it's amorphous. It's a blob that second to second, minute to minute, changes shape. These amoeba utilize what is referred to as pseudopodia. Pseudopodia, remember, rule of thumb, don't know the word, but what it means, break it into its parts. Pseudo means fake, podia means foot, fake foot. What happens is you have the whole cell. It's just sitting there doing its thing. All of a sudden, part of the cell stretches out. The rest of the cell flows into that. And it just keeps doing that over and over as it's moving towards or away from something. Original microbiologists thought this looked like it was taking steps in some weird way. Hence, pseudopodia. We're also going to see flagella here. Now, previous lectures in this chapter, remember that flagella and the cilia here are completely unlike what we saw with bacteria. Okay, flagella, cilia are going to be internal structures that push that plasma membrane out. The cilia going to move like this. One cilia moving doesn't do much because they're very small, but you get hundreds and thousands of them all over the cell surface doing this. Well, it starts to move a lot of water. You move a lot of water around the cell, cell swimming. Same with flagella. Flagella is still inside the cell, but now it's moving like this. Instead of that spinning around thing like the bacteria do, imagine if you were swimming in the pool and you're kicking with your leg. That's exactly the motion you're going to see with a flagella, just a lot faster. Now, we generally don't utilize shape as one of our criteria, because we're going to see in this lecture, the shape of the protozoa can change. Many of them can change depending upon what's going on in its environment. You look here. This is Euglena. Euglena shows us, well, they kind of sort of all have the same shape, but the cell is not rigid. It can bend. Cytoskeleton can make it so it bends and moves as it's swimming along. You can just barely make out. Right in the middle of all of them, you see some light lines. Flagella. Great picture here also shows you the difference between ectoplasm and endoplasm. You see parts of the cell where it looks granular, but there's really not much there. Other parts of the cell that seem to be packed full. Here the Euglena is packed full of little green spheres. Those green spheres, chloroplast. Now when it comes to nutrition, turns out protozoa are the medically important ones, the medically important ones are heterotrophic just like we are. Think about it. If it's photosynthetic, it needs the sun so it can stay alive, it can make sugars, whatever. Once it's inside of you, no sunlight. The cell's going to die off faster than you will. So heterotrophic, like us, has to take in its carbon sources from outside, its energy sources from outside in. Habitat for protozoa, everywhere, as long as there's some moisture. Now, it doesn't have to be lots of water. Yes, these things are going to be in streams, lakes, rivers, ponds, the ocean, but there are also going to be some in soil, as long as the soil is moist. Whether it's, rains once in a while, or it's near a waterfall and it's constantly getting misted. Remember, the size of these protozoa is so small, they can literally swim in between soil particles. As long as there's moisture there, they're good to go. Now we're going to see that for many of these protozoa, they seem to fluctuate between two stages in their life cycle. Trophozoite, the feeding stage, where you see, this is the cell you see pictures of, the amoeba as it's flowing across the slide, or tetrahymen and things as they're zipping around, swimming, feeding. Signal comes through the trophozoite because of running out of water, running out of nutrients, pH changes, so many different things. It's going to switch from trophozoite to the cyst form. This is a resting stage. The cyst is nothing more than a protective coated cell. It is not an endospore, okay? When you look at the cyst, you're going to see that a trophozoite gets the signals, external signals, whatever they may be, and it starts laying on its surface, coating and coating of proteins and sugars on the surface of the cell. Not a separate cell inside of it like an endospore. And these coats are very thin, okay? It's, can help protect against drying out to an extent. It's not like the endospore that's not bulletproof, okay? Endospores can dry and then be stable for years, decades, centuries. Cysts like this, not so much. Boiling these kills them. Wouldn't kill an endospore. So this protective coating is just there to protect short term. It's not, the be-all, end-all. Water comes back. Whatever the signal was goes away. Cyst walls split open. Out comes the trophozoite. Now, when it comes to reproduction, it's going to have to be the trophozoite cell. The cysted cell is metabolically inactive, so it's just sitting there. So the trophozoite is going to divide. Turns out protozoa are mostly asexual reproducing organisms. They go through mitotic division. Yeah, mitosis. You remember that. Prophase, metaphase, anaphase, telophase, cytokinesis. Same thing here. So each successive mitotic division will give a genetically identical offspring cell, which is not to say that there is not some sexual reproduction occurring within the protozoa. Not all of them can do it. Some of them can fuse together. They fuse together, become one giant cell, mix the nuclei, split the nuclei, and out comes two cells that are still the same species, but have now genetically recombined. Or you'll see what's called conjugation. Two cells kind of fuse just a bit, swap DNA back and forth, and off they go. That's the protozoan version of sexual reproduction. When you look at, who causes what when it comes to diseases, we'll get more in depth in these at the end of the semester. But you see, there are still quite a few protozoan diseases out there. Some really, ones that are highly prevalent. Entamoeba histolytica. You drink water that's been fecally contaminated. You eat food that was rinsed with water that was fecally contaminated. You're going to get Entamoeba histolytica in your large intestine, and, well, you're going to have diarrhea for about a week. Or, on the flip side, you can go swimming in a pond, swimming in a stream, swimming in a lake. What you're going to see happen, water gets up your nose. Naegleria fowleri or acanthamoeba are going to attach to the olfactory nerve, and they're going to swim up the olfactory nerve to your brain. You're dead before you know it.